Next up, I'd like to invite Chuck. So Chuck is also one of our ACD fellows. Chuck received his PhD in medicinal chemistry and molecular pharmacology at Purdue University in the lab of Dr. Eric Baker. He uses computational biology approaches to understand the mutational sequence constraints of membrane proteins and how this contributes to disease. This has relevance for SLC6A8 or the transporter protein that is implicated in CTD, since it is also a membrane protein. Today, Chuck is going to tell us about his studies searching for possible small molecule therapies for creatine deficiencies using virtual screening approaches that involve molecular docking and protein structure prediction and modeling.
Thank you. Can you guys hear me? Okay, great. So first of all, I'd just like to thank ACD for inviting me to this and for all the support over the last couple of years. You know, it's really fascinating being here. I've met some of you, and meeting you guys is just really motivating and inspiring. And so I'm very excited to continue this work. So, you know, there's some therapies that exist if you're able to identify it fast enough for AGAT and GAMT mutations. But right now we don't have any therapies for people who have SLC6A8 variants. And so we're working on that using the principle of the idea that some of these variants are going to affect the folding of the protein. Now, some of them, as you can see, if you look at this graph, you'll see that we have some variants on here. And basically, each point is a different variant. And we recently did a study. There are over 80 loss of function variants for SLC6A8. And some of them impact function by direct impact on the binding site. That's what those two variants, G421R and P382L, those will do that. And the rest of the variants should fall on this dotted line where you can see that it's a graph of creatine uptake, which is a proxy for function, versus plasma membrane expression. So when the gene is synthesized, it's synthesized in the endoplasmic reticulum, and then it's trafficked to the cell surface. And proteostasis defects caused by variants will actually impact the biosynthesis of the protein so that it can't traffic to the cell surface. But in cystic fibrosis, we've seen that some of these variants are such that if you can actually use a small molecule corrector to reach the site of biosynthesis, you can actually increase the trafficking and the fraction of native, the fraction of folded protein so that it can traffic to the cell surface. So in cystic fibrosis, we have a concept of a potentiator which can basically correct for these kinds of defects like G421R that impact function directly. And then there's correctors that can repair defects that affect the native fold of the protein. And so my studies are a new way, I'm trying to find a new way to screen for correctors by targeting a novel binding site. So, you know, we're going to leverage the fact here that SLC6A8 is an SLC6 protein. Okay, so this is a protein that is very closely related in evolution and structural biology to one of the most studied targets in pharmacology. So we have all kinds of structures, a lot of drugs bind to SLC6 proteins. And we're interested in finding drugs that specifically target SLC6A8, the creatine transporter. So, you know, the transporter uses this alternating access mechanism. This is an old paradigm, but recent X-ray crystallography studies and cryo-EM studies have really validated the alternating access model where, you know, you'll have the creatine or some substrate from on the outside of the cell come in along with sodium. This is a sodium-driven process. So there's a sodium gradient across the cell membrane. There's not a sodium gradient across the ER membrane. So what will happen is that the substrate will bind to this outward-facing conformation. There's a conformational transition, and then the substrate is basically being able to uptake into the cell. In the case of the creatine transporter, this is creatine in neurons. And so what we want to do is we want to actually target this inward-facing pathway because in recent years there have been cases where Wellbutrin, an antidepressant, and Ibogaine, another drug, have been found to rescue the expression of SLC6 variants, in this case monoamine transporters. But are there equivalent small molecules that can target the creatine transporter. And they do this because in the ER, it's thought that the sort of default state of the protein is in this inward-facing conformation. So if you can target this inward-facing pathway specifically, basically this cavity that you see that's pointing down into the cell in the ER membrane, then maybe some of these small molecules will be good candidates for correctors. And so we don't actually have any structural information directly about the creatine transporter, but as I alluded to, we have a wealth of new structures of very closely related proteins that we can use to build high quality homology models. We also have AlphaFold now that we can even tune AlphaFold to predict the structures of specific conformations of the proteins. It's a little bit tricky to do that, but you can do it. So yeah, we designed a counter screen, and basically what we're gonna do is we're gonna do a virtual screen against the inward-facing conformation. We're gonna take those same compounds and dock them to the outward-facing conformation. We're gonna estimate the binding energy for both conformations. We're gonna basically look for the compounds that have the most bias towards the inward-facing conformation. And so, you can see here some examples of some of these proteins in the intracellular permeation pathway. You can see creatine bound here. This is all based on our model, but these are all based on templates of very closely related inward-facing structures that have come out in recent years. So 3TT3 is from the leucine transporter. That's the oldest inward-facing structure. We also have new structures of the serotonin transporter in the inward-facing conformation and the glycine transporter in the inward-facing conformation. You can see that while there's some variation in the movement of helix 1, which is sort of the hinge helix that opens up on the inside to form the inward-facing permeation pathway. You can see that there's some variation in that, but where the creatine actually is, it's pretty conserved even across these three different structures. So we started out by validating this method on a set of known metabolites. So Randy Blakely, when he first characterized the gene for SOC6A8, looked at all these metabolites, and he found this 3-guanidinopropionate and gamma-guanidinobutyrate and a few other compounds that are actually very close analogs of creatine. And you can see that the main chemical groups of creatine are pretty conserved across most of these. He also looked at a bunch of amino acids that aren't so close, but they didn't have very much activity. And so we tried out this approach by docking all those metabolites that have known activities for the creatine transporter. And we looked at that measure of bias that I was telling you guys about. And you can see that the compounds that have an outward-facing bias, they have sort of diminished activity. So this is indicative of the fact that some of those compounds are actually inhibitors. Obviously, we don't want any corrector that we develop for the creatine transporter to inhibit creatine transport activity. We don't want it specifically to work in the ER. But you can see that the compounds with an inward-facing bias actually had no impact on the activity of the creatine transporter. And so moving on to our screen, as I mentioned before, we docked we basically used data mining approaches to mine 9 million different compounds for creatine and 3GP analogs. And then we basically used those to dock to both the outward-facing state initially and the inward-facing state initially, because we wanted to kind of compare to see if compounds that fall out of an outward-facing first screen would still have compounds that when you dock them to the inward-facing state that they would still work. And of course, we did the same thing for the inward-facing state. We found a bunch of compounds that bound to the inward-facing state, we redocked them to the outward-facing state, and then we looked at their activity. So you can see on the right, this is called a violin plot. It's basically just a distribution of the binding energies of all these compounds. So for the blue is going to be the scores that are docked to the outward-facing conformation, and the red are the scores docked to the inward-facing conformation. You can see that for our inward-facing first screen, that you can see that they have very good affinities for the inward-facing conformation. Not so good affinities for the outward-facing conformation. Far more molecules fell out of the outward-facing screen initially, but you can see that a much smaller percentage of them have favorable activities to the inward-facing screen, but we still got some hits. And so looking at the individual energies of these hits, you can see that you have this distribution of different compounds. The green circles are the ones from the inward-facing first screen. The gold are sort of the outward-facing first screen. And you can see that what we're really looking for here are ones that have unfavorable binding to the outward-facing state, but favorable binding to the inward-facing state. These are going to be our screens. So in this case, the more negative the number is, the better it is, because these are binding free energies. So this is the one that we don't want. This is one that has a very unfavorable inward-facing binding energy, but a favorable outward-facing binding energy. And so what we did was we did this, and a lot of compounds fell out of our hits, and you can see some of them here. These all have pretty good log P's. So what log P is it's a measure of the hydrophobicity of the compound, and it's basically a measure of how drug-like it is. And so you want log P's between 1 and 4 for a lead compound, and then they usually get more hydrophobic with development. But you can see that they all contain either the creatine or 3GP in their substructure. It might be tricky to see that in some cases, but it's all there. And you can see that these all have very favorable inward-facing biases. So the difference between the inward-facing conformational energy and the outward-facing conformational energy is quite negative in all these cases, which is a good thing. So looking at how they actually bind, we have some studies from our previous work and from other investigators in the field about sort of how creatine binds to these transporters. And these, starting with creatine and 3GP analogs was good because it allows us to do a sanity check and make sure that the binding poses that we find sort of line up with what we already know. And in this case, you can see that, but what's new is that they all have these bulky chemical substituents just in the right place to sort of wedge down into the intracellular permeation pathway. And we think that that's really what's allowing them to lock the transporter in this inward-facing state, which hopefully means that they'll get into the ER and bind to the protein and shift the balance in favor of this site. So we also have some promising compounds from our outward-facing first screen, but if you look at the ones that actually were successful from that screen, they look very similar to the compounds that fell out of our inward-facing screen. So we got lucky in this case, and they have the same properties where they have these bulky chemical substituents that sort of go down into this intracellular permeation pathway, whereas the creatine part of the molecule sits where creatine normally would. That's all very good news. And so we're ready to basically do experimental characterization of at least 55 of these. We actually just finished another screen where we have at least another 100. So we should be able to easily do a 96-well plate for Axum to help us characterize. So we're basically ready. You can see basically the distribution of all these compounds here. That delta G is basically that bias score that I was telling you guys about. The more negative it is, the more favorable it is to the inward-facing state. You can see that for the inward-facing first screen, that most of them, were good hits. For the outward-facing first, we still had some good hits, but more of them were not favorable for the inward-facing state. And then we found some compounds that fell out of both screens, and you can see that those were mostly favorable. And so, how do we take this going forward? We have, you can imagine this space of all possible small molecules, and somewhere in that is, compounds that bind the outward-facing state of the creatine transporter. You also have compounds that bind the inward-facing state of the creatine transporter, and compounds that bind both, compounds that have favorable binding to both the inward and outward, those are actually substrates. So ATPCA is an example of a partial substrate for the creatine transporter. We also have creatine, of course, is a substrate. It's going to do this. It's going to bind both. What we want is a compound that's as far from the outward-facing state as possible, a compound that really selectively binds this inward-facing state and rescues expression at the cell surface for the largest possible number of variants. And so going forward, what we want to do is what we've seen basically is that prioritizing inward-facing based compounds may help avoid the discovery of inhibitors. We've identified at least 30, possibly as many as 50 potential new pharmacological chaperones. Of course, that's all very contingent upon experimental characterization, which is the next step. So now what we can do now, once we get a lead compound based on one of these hits, we can basically use that and do that data mining technique that I mentioned previously and look for analogs of those and basically repeat this screen using a better starting molecule. So as we do that, we'll slowly be able to get, well, hopefully not slowly, but we'll be able to eventually get a really, really powerful corrector for the creatine transporter in the way that we have correctors for cystic fibrosis. And so then we're going to work with our collaborators at the University of British Columbia, one of whom you're going to hear from during this symposium. And apparently also we're going to work with Axum. So I'd like to thank the people in our lab who've helped with this, especially Marty, who sort of initialized our studies on the creatine transporter. And I want to thank Yulia at the Simons Foundation for sort of helping us get started with modeling the creatine transporter itself. She built our outward-facing conformation of the transporter. And I want to thank our collaborators at the University of British Columbia, but I especially want to thank ACD for their very generous support over the last couple of years. And with that, that's it.
Thank you, Chuck. I'd like to invite our previous speakers as well as Dr. Longo. Leah, you can take a seat on the podium. So Chuck, Alex.
I want to thank all of the speakers for their succinct and very clear presentation. And what I would like to do is try to highlight a second how all of this research can really be used to improve the life of people with with brain creatine deficiency syndrome. And I'm going to ask a few questions, but questions from all of you are really welcome. And my first question for Mr. Lee here, who did a very good job in showing how to repress the expression of the AGAT gene. Obviously, if we can fully inhibit expression of the gene, we would limit the synthesis of guanidinoacetate. So patients with guanidinoacetate methyltransferase deficiency could be used a less strict diet and less amount of supplement. And my question is, how much with the best possible scenario can you inhibit expression of the AGAT gene?
So essentially, you're essentially asking me how much can I repress AGAT, basically. Well, based on the research that I showed, using just the minimal peptide sequence that we have, we were able to repress it anywhere from around 50% approximately. But if we look at the full-length gene itself, some work in our lab has shown that you can press it up to 90% expression. So there is quite a bit of variable depending on how much of the AGA gene in question you have.
90% would be a very good achievement if achievable, because obviously that would be really nice. So, and then we had a beautiful presentation by Dr. Chuck Kuntz, who presented a lot of chaperone that could restore activity of the creatine transporter. Obviously, you take this medication, and usually the creatine transporter is broken down because there is a genetic mutation. What the chaperone would do, it would bind to the transporter while it is being synthesized in the endoplasmic reticulum, and the endoplasmic reticulum would be able to transfer the transporter to the brain. So obviously this has been achieved already in cystic fibrosis, where the new polypharmacology is able to increase dramatically activity of the cystic fibrosis gene. The cystic fibrosis gene is another membrane transporter, exactly like the creatine transporter. My question for him is a little bit different. I'm a functional guy, so how do you measure creatine transport? Which cell do you use to test efficacy of your compound?
Well, when we have an assay that we actually do lots of mutations in parallel in our lab. So our lab is really interested in membrane protein membrane protein homeostasis generally. We call it proteostasis for short. And we actually do deep mutational scanning studies on a number of different proteins involved in dysregulation of membrane proteins. We do studies on CFTR. We just actually had a paper published on CFTR where we do a pharmacological profile of many different variants of CFTR I think that it would be really cool with some of these compounds to do the same kind of thing on the creatine transporter. And I think with the work that we're doing in our lab now, I think we're going to start ramping up our work on the creatine transporter. We've also done a similar pharmacological profile on rhodopsin. So our assay is definitely an in vitro assay to actually, really demonstrate conclusively that you improve function. We would need to collaborate with people who have, who work directly with cultured neurons, for example. But our assay is really designed to measure the trafficking to the cell surface. And we use sort of a model system for that my PI, John, developed for this purpose. And that assay is done in HEK293 T cells. But we use that assay for all of our sort of massively parallel studies of many different variants for various membrane proteins. And it works well for measuring expression. And so in the search for a corrector, that's really what we're after. We're after, looking for things that restore expression at the cell surface. And then once we find something that can restore expression at the cell surface, that will take care of some variants. There are going to be other variants that's not going to help, because there are some variants that have a loss of function, not because of their impact on the global fold of the protein, but on some direct impact on the binding site. And so once we have demonstrated that we can restore expression at the cell surface, then we pass it along to our collaborators to really demonstrate that function is restored.
Thank you. Let me translate a few of what he has said, okay? So basically, what you have that many of the mutation in the creatine transporter gene retain the transporter on the inside of the cell. It cannot make it to the surface of the cell. What they are screening for compounds that can bring the transporter to the plasma membrane where it is supposed to be, on the outside of the cell, not on the inside of the cell. So, and once they achieve that, they are able to, they will pass the compound to one of their collaborators who will test for actual activity. Now, obviously, sometimes you have mutation in what we call the active site of the transporter. Let's say that there is a mutation in the site that recognizes creatine. Obviously, if you do not recognize creatine, even if you bring that to the plasma membrane or to the surface, the compound will not be able to work. Many times, people have been donating fibroblasts to biobank, and my question for him is, could we test your compounds in fibroblasts from patients to see if they restore activity?
Sure, I think that would be great.
So the answer is it would be great. So in other words, if you have in mind, or if you have available fibroblasts, sharing them with the scientific community can increase our understanding and the development of this compound.
Say one quick thing about the compounds that I found. They're all we can purchase. They're all readily available. They're not. They don't just exist in the ether. These are all compounds that are ready to purchase on the shelf, off the shelf, that we can test directly now. So we wouldn't rely on some medicinal chemist to spend a year synthesizing them. We can buy them.
There is a question from the audience. If one can pass a microphone back there.
Sorry, falling behind on my job.
Faster, faster.
Get my steps in.
Thank you. My question is for Dr. Kuntz. Thank you very much for your impressive talk. And you show two promising analog creatine. The first with a lipophilicity about 1.17, and the other one minus 0.3. Do you have some results on PK properties in terms of membrane diffusion of those two compounds?
No, we haven't actually done any experimental work on these compounds at all. These results are sort of hot off the presses from our virtual screen. And so the next step that we're going to initialize basically this, over this summer, we're going to hope to get started on this, is we're going to pick some of the best, most promising compounds from this We're going to purchase them, and then we're going to do things like testing whether we can get them across cell surfaces and whether they can actually be used for this kind of thing. And by the way, I will point out that those log Ps, they're called A log Ps because that's a really good method for estimating log P. What a log P really is, it's, you take oil and water, you put the drug in this oil and water mix, and you see which percentage of the drug goes to the oil and which percentage goes to the water. That hasn't been done for all these 9 million compounds that this virtual screen came from. So A log P is basically looks at the chemical structure, and it gets a really good estimate of the log P, but it's not, it's not perfect. So we actually have to determine that stuff experimentally before we proceed with before we can answer a question like the one that you just asked.
Dr. Schulze.
Yeah, for Charles as well, question. So you're basically, you have now a lot of experience with doing these kind of experiments to identify potential molecules. But there is one thing that I would think is specific to the creatine transporter that I would recommend you should include into your early screening docking experiments, and this is the off-target effect that those molecules could have to creatine kinase. And why I'm saying that? So you have, for example, beta-guanidinopropionic acid and metabolites as perfect candidates. They are well known that they bind, but they are toxins. Why? Because they bind to creatine kinase, and the most effective cell toxins. And of course, you can expect any kind of creatine analogs that bind somehow to the creatine transporter, that they could also bind to creatine kinase. And in order not to avoid going too far with molecules that at the end of the day turn out to be toxic, I would put this in the first line of screening.
That's a really great idea, and that's exactly why that's the kind of remark that I was really hoping to, the kind of conversation that I was hoping to have here. So, this whole approach is sort of a counter screen. So we dock to inward-facing site, and we have a counter screen against the outward-facing site. Incorporating a counter screen to known targets like the one that you just mentioned would be really easy and a really great use of our time to incorporate. So thank you for that suggestion. But I will point out that a lot of those compounds, some of the analogs do look like they could be phosphorylated, and other ones don't. Other ones are acetylated in various things, and that has its own issues. But hopefully, some of the compounds on our screen might bind to inward-facing creatine transporter, but not bind to some of the other targets that you mentioned. But I think that that's a really great idea.
Dr. Leah Broca-Brisson presented beautiful data on organoids. Organoids are a way of studying the brain without actually having a brain. And the advantage is that you can basically build a brain with creatine transporter deficiency and then see what happened there. What she found that by studying proteins in this different area of the brain, she found that several proteins involved in intellectual disability are dysregulated, and that obviously could open the possibility of modifying the expression of the gene encoding this protein to alleviate the symptoms of some of the symptoms that we see in creatine transporter deficiency. And I'm asking her if she has any idea of how that could be possibly achieved. The question is, you identify several proteins that are upregulated or downregulated in the organoids of patients with creatine transporter deficiency. Are there ways of modifying, of correcting the profile that you see? Or can you see one?
Well, we can try some screening of drug candidates known to be involved in the pathways that we're going to identify and link to the 32 proteins. And maybe we're going to identify potential drug candidates that are going to rescue the dysregulations that have been identified on CTD pathophysiology.
Thank you. So having a target obviously would help us determining what we need to correct.
I was just going to ask, I mean, it's amazing that you're doing basically the test brains there. Will we be able to test drugs on those test brains there without?
Yeah, this is the last objectives of the thesis of my PhD, is to use them as a tool to test the efficacy of drug candidate. So it's the second part of the work that I'm going to conduct.
Awesome. And since I'm here, I'm going to ask one more, if you don't mind. to Dr. Kuntz, I appreciate your work there. It sounds like that there may be some compounds that exist now that are, I guess, being uptaken for creatine and transported. Because could we be getting some of that in our normal diet? And that's why we're seeing some variation in the CTD patients.
That's a really interesting idea. I'd have to look for studies that sort of systematically look at that, but there's all kinds of phenomena that, things that we can't explain in medical science. And, COVID was a perfect example of there's all kinds of unanswered and even unexamined variation that we see in patient outcomes that COVID, because it was such a massive event, people started seeing things that they never really investigated before. And I think that that's a perfect example of a line of investigation that would be really interesting to see, like what kinds of even things in food that people eat and metabolites of other drugs that they take for various things, could have some small impact on these things. And so I think that's a really interesting idea.
Thank you. I think that our time is over. The machine is going to zap me if I go beyond. So I wish you a good coffee break. But first, I want to thank again the speaker for their outstanding work. Thank you.